Welcome. Great. Why don't we take a quick minute just to sort of go around, introduce ourselves, and maybe if there's a specific question or sort of idea that you have, I'd love to hear it. And I'll also kick it off. I'm Rich Schweitzer, the academic director of the Word and Behavior Lab. My goal is to make the Word and Behavior Lab an extremely good place to support research. I'm keen to build bridges with Word research computing to figure out how we can support the research endeavor here at Word. So I'm eager to hear ideas that you might have for research and support whatever research you're doing better.
Hi, Alec Wan. I'm the executive director of research analytics at Word Computing. So Robert's part of the team, and I work a lot with Mary and Maurice and everyone else. Thanks a lot and support them with technology.
Hi, I'm Max Gary. I'm a postdoc in the marketing department here, doing CD research, working with Robert on a couple of different projects, always interested in new methods, because I think there can be a USP in the job market or in the academic field in general, I would say. And I'm mostly interested in attentional mechanisms, so therefore I'm always interested in how can we actually capture them. And eye tracking is one way, but mouse tracking is another, and so on and so forth. So that's why I think it's still underexplored. I think what used to be much bigger in the past, but I wonder why is this? But,
Hi, I'm Lee. I'm a grad student in psychology and marketing, working with this Brown and Michael Black. And I have a experiment that I'm using eye tracking and implemented is related to numerical cognition and also related to decision-making because I will get like beauty and more.
Hi, I'm Liz Beard. I'm also a postdoctoral research fellow with the Word Neuroscience Initiative. So working with that and Michael, and we have a couple of projects that will be starting that are looking at like visual attention processes and different ways to kind of like dynamic video stimuli. So I think that's what we're focusing our efforts now.
I'm Zab Johnson. I'm the executive director and senior fellow for Word Neuroscience, and I teach visual marketing with Barbara Kahn. And so I'm a longtime user of eye tracking in the WBL. So I'm just here to help support and see what I can learn.
I am Tom Stewart, Senior IT Support Specialist in the Word Behavioral Lab. I assist with the hardware and software support with the lab.
Mary Spratt, I'm the Associate Director of Behavioral Lab.
And I'm Robert Fotto, IT Director, is that right? Something like that. Word and Behavioral Lab. So let's jump into this. We're going to talk about eye tracking. We'll start very generally as like an introduction. We'll go through that quickly and then talk about some of the devices and things we have. I might stand while we do this. So let me select my PowerPoint. There we go. A little cut off at the top. We can live with that, I think. So why use eye tracking? What can we investigate using eye tracking? First and foremost is attention. So attentional measures, attentional mechanisms within decision making, research you might do. So we'll talk about that first. We're gonna kind of use this as a guide to walk through some of the methods and visualizations and things that you will use. Second, reading and comprehension. There's reading is itself a kind of subfield within eye tracking, and then also arousal and engagement method or measures and cognitive load or effort. So we'll start from there as our framework. So for attention, right? So attention is one of the earliest and most common uses of eye tracking. So if you look at the human visual system, probably most people in this room know this, but vision is a highly selective sort of filter of the world around you. You really only can bring in and process high acuity information in about 2 degrees of the visual field around you. That's about your thumbnail or quarter held out at arm's length. So you're sampling these sort of tiny spotlights of information approximately 4 to 5 times a second. That's sort of our model of how vision works. So if you, and so that's corresponds more or less to where the cones are in your retina. This is sort of a diagram showing those. This is sort of the field of got this. So from the field of vision, say, that small blue line is high acuity. It then drops off very quickly at about 5 degrees each side. We call it the sort of parafoveal area. And then periphery is where is rods. That's just like big movements, fight or flight kind of wiring.
That's a peripheral vision.
That's the peripheral vision, yeah, which is like generally not associated with attention. I mean, it is, you can sort of coerce yourself into paying attention to what's going on, and sometimes that's helpful because they're actually more sensitive to low light. So like astronomers use their peripheral vision to see distant stars and stuff, but generally attention is associated with foveal vision, where your eye is moving very quickly. So how can it be that you seem to have a very clear mental image of the whole visual scene around you? It seems continuous and complete, but it's really, and we can't, I'm not qualified to really get into the details of how that works. We'll just say that our brains are very good at assembling this whole and continuous mental image using these piecemeal little samples of highly detailed information, including cues that come from the outside. And so there's whole models of how attention is guided then based on, say, parafoveal stimuli as well. So you might start to anticipate why it is that eye tracking is attractive to research, because if you can accurately measure where a person's gaze is fixated, then you can build a map and see what's salient, what's important, what's drawing attention for visual processing. Because ultimately, if the eye doesn't fixate on something, that information is not processed. It doesn't make it into the brain. So it's a real, like I said, a real filter, I guess, of information. So you can see what details are attended to versus what was skipped over entirely. You can also look at the order in which somebody looked at things, like scanning, rescanning visual stimuli. These could be natural scenes, video, photographs, product packaging, other more abstract user interface controls, written text, all of that. Here's an example of a common visualization you'll see in eye tracking. This is a heat map. So heat maps could correspond, say, to attention. In this particular example, this is an advertisement for dog food. You wouldn't necessarily get that right away, or pet food, I guess, in general. A few things to take away from this. Faces. Oops. Faces draw attention. I think there is, Zab might know this better, I think there's actually mechanisms in the brain that are purpose-built really to identify faces other than non-face objects. Like they really get priority in our attention. So always, but then also, of course, in this case, an advertisement, you have your message and stuff up here. You've got something going on here. I find this ad interesting. It's a little puzzling what the connection is, but anyway, we'll move on from here. So and by the way, this is 12 individuals watching this. This is an aggregate of data collected over 12 individuals.
Yeah, I was gonna ask about it.
Yeah.
So like some of us, like you are sort of an expert analyzing, collecting all this data. But for those of us, I guess more like Max, like not expert yet. Like that's ever. Yeah, how do you sort of get the data for this, and then how do you use that heat map to sort of then make the inference?
Good question. There's whether heat maps are really useful for analysis beyond a sort of illustration that would then lead to a more rigorous analysis, I think is a good question. It's a good starting point, but I do think that you have to go a little further. We'll get into a couple of different ways to do that, but yeah, I think that these are helpful guidelines for attention, but aren't that useful for actual analysis in most cases. It's hard to like translate this into statistics you can really like talk about.
It's an easy visualization.
Yeah.
Are you gonna show a scan path too?
Well, 2 slides from now, we'll get to a scan path.
Yeah. So I mean, I like to think about
That's
these
what you
as
do.
really
Yeah.
straightforward visualizations, but they're not analyses.
Yes.
So they're a way of sort of binning the data in ways that then you like one could look at and think about
Yep.
how that might guide further.
One thing to think about that data, I mean, that's gaze points mapped to basically pixels on a screen, which isn't that useful. You kind of want to, you could bin up from that, and we'll talk about that to start to get more insights. So once you get away from just attention into maybe comprehension and reading, you want to focus more on the order of fixations, draw conclusions about how that information is processed. That might be based on assumptions about reading, left to right, top to down, or just previously observed movements. Did the eye move left to right and then back to right? So you want to look at these, and there's different measures to describe that. Backtracking is sort of moving directions. Regression comes from reading. It's about sort of rereading sentences. Looking back, looking ahead is somebody scanning very broadly with a wide dispersion, or sort of searching really tightly within a scene. And of course, this is all intertwined with attention. Here's another visualization, one that Zada just mentioned. This is what we call a scan path. So here we've sort of abstracted those fixations into these circles and plotted it on top of our stimulus. And it's kind of hard to see, they're numbered in the order in which we recorded them. The larger the diameter of the circle, the longer the fixation. The straight lines in between are an abstract representation of what we call a saccade. That's the very quick movement on the order of tens of milliseconds between fixations. It's important to note that you don't process any information during a fixation. You're basically blind when that's happening. So you're really only processing the points where the eye stops. So here, for example, face is really important. This is one person, by the way. This is just one individual. Looking at the face, we'll use the pointer here, up to maybe the ad, looking down. You can sort of see like what's somebody puzzling out what's going on here. They return to this multiple times after coming down to the scene. You probably can't read what it says, but what it says is company for the rest of the year. So I guess it's about maybe under the table, a pet who's hanging out and is there on days that isn't this guy's birthday. A little sad, but that's the sad.
That's very good.
It's a pretty good one. But I think this tells a lot more. I think this tells more of a story than that heat map. I think you can get a lot more into how somebody's processing this information. But this is for an individual. So now you want to think about maybe how you'd aggregate that up, and we'll get to some of that. So the fourth bullet I mentioned earlier was arousal or engagement. So along with gaze position, eye trackers usually report pupil measurements. This kind of comes for free with the algorithm because the camera in the eye tracker has to find a pupil to begin with. So measuring changes in diameter of that pupil is sort of part of the positional algorithms anyway. So, but far from an afterthought, changes in pupil diameter reveal a lot about mental state. So for one, if you're doing like affect research, an increase in pupil dilation can indicate arousal. If you're doing maybe human factors engineering, it can indicate engagement in something. So actually a lot of the research we've run in the lab focus more on the pupil response, even than positional data. So, but pupil dilation is also observed during high degrees of mental effort or cognitive load. You can reliably test this doing end-back memory tasks or just mental arithmetic calculation. And that will cause people to dilate. But then again, so will arousal. We haven't even talked about light reflex, which is the overwhelming cause of change in pupil diameter. So to draw inferences from pupil measures, you really have to be very careful about your experiment design and really control to mitigate all of these confounds. And this is a fun illustration. This is a study done. These two stimuli that differ only in that one looks like a sun and one has a picture looks like a moon are isoluminate. In other words, they're the same brightness, but eyes will contract more on the image of the sun. So there's high level stuff happening to this. So there's a lot going on with pupil diameter. It's really important to control for all these things.
Is that really use pupil dilation a lot?
A lot.
A lot, yeah.
And it's emerging as being like an indicator of an early indicator of like stress resilience, for example. Now people are like, I just read a paper this morning, actually, that was on interventions, but like using pupillometry as biofeedback so that people can actually learn strategies to mitigate their own stress responses, for example. You just have to be really careful because as Robert said,
It's a single number that encodes
Yeah.
so many potential causes, you know, and so, yeah, you have to be very.
so anything on the screen, but also anything in the ambient environment can also affect pupil diameter, so to be really careful that you know what's really causing pupillary changes.
So how to sort of like break it down, so you get pupil dilation or diameter. You get sort of where they're gazing for how long, and
Yes.
the order in which they're gazing at different points.
Yes,
And then, so like that's the data set. Right?
more or less, yeah, I'll show you like some
Yeah.
examples of that soon. But yes, you're getting there. That's the original kind of raw data set. Here's an illustration of a couple of eye tracking systems. They come in all shapes and sizes these days. So top left here, this is actually one that we currently own. This is a Tobii Profusion. This sort of adheres to the bezel of a computer monitor. There are wearable glasses. There are huge room installed towers that limit head mobility, that usually operate at a higher sampling rate, that usually have higher accuracy and precision specifications. So there's trade-offs between all of these. So yeah, eye tracking systems, they vary in scale. They can be lightweight enough to be built to wearable glasses. They can require permanent installation. These days, most are in the middle. They attach to your computer display. They're placed in front of a subject. We may or may not restrict head movement. We do use chin rests for that sometimes. And then recordings are then synced with stimulus display systems that also record onset times, user feedback, whatever the design is of your study. And that's in roughly what an eye tracking system is. Here's an illustration from Tobii of some sort of a laptop mounted eye tracker. So I guess going through the numbers here, eye tracker will be attached to a monitor here. It has cameras, it has the lights, because it uses IR light, infrared light, not visible light, to reflect off the eye. It then uses that reflected light to identify the pupil and also reflections in the cornea. Then it has its own internal algorithms that do machine vision to actually identify those landmarks, and then do the calculations required to, say, map eye movements. And that's, of course, all happens in the computer. So kind of as we just said, bottom bezel of the computer monitor, that's IR cameras. Here's, I think that's my eye, a picture from one of our older systems. See, oops. Here, a crosshair in the center of my pupil, and then crosshairs on 2 or 3 corneal glints that it picked up. And so we will use images like this in real time to help with data quality and collection. So one of the first things to do is when positioning a participant is make sure that they're positioned relative to the screen in a way that the eye tracker has a good view of their eyes. We use these kinds of visualizations for that. Our old eye tracking system, this is what we have the most experience with. For about 10 years, we used these SMI Red M eye tracking cameras. Everything I said applies to them. Infrared lights, cameras, they record at 120 hertz or 120 times a second with an accuracy of about half a degree on the screen. And then stimulus presentation is handled by software from SMI, they call it Thermo Center, and then also they have a software for doing analysis. Our newer systems, we just bought one from Tobii, Tobii Profusion. Fundamentally, it's not really any different from the old one. It's a little faster. It can record at 250 hertz. It has slightly better accuracy. One interesting thing about the Profusion is it can do both dark pupil and light pupil, and it actually can, what that means, most are dark pupil. It's just the infrared cameras light up your eye. It's not like an inverted, it just looks for a dark pupil. Under certain lighting conditions, it's actually, it might be advantageous to, if you bring the infrared lights closer to the camera, you can sort of wash out the pupil. It's similar to like the red eye in a flash from a camera, if you remember those days. So then it creates a light pupil, and that under some sort of conditions can create higher accuracy. So it can dynamically change between them, which is interesting, that you are limited to 120 hertz if you're going to use that dual pupil mode, but something new. This is me. So this is more of the reality of a setup. So what we do is rather than having a single display setup, we usually have a laptop that an RA would be looking at, and then a display just for the participant. We do often use chin rest to control for head movement. And the eye tracker itself is this bar here attached to the bottom of the screen. So this is what the eye brochure shows you. This is more what a real setup would look like. This is the Tobii system. We also have a Smart Eye Aurora. It's very similar. This one, the big difference is the software that it ties to for data recording, for stimulus presentation, and for analysis. It's really tied to the eye motion software. Zav and other people have had some experience using it. Very similar specifications to the Tobii, with the exception that it's only dark people. Here's that setup. It's a bit more cluttered. It again, here's the sort of ad brochure version. This is more like reality. Comes with something that euphemistically call an EPU, which is literally just a computer that it attaches to, and then that computer attaches over Ethernet to our other machine. A little bulky, but it offloads some of the processing power here. We'll see in the coming months as we use these more, how advantageous it is to have the extra processing, handling eye tracking, not sharing, say, the computer's processor replaying video or something like that.
And how many machines do we have right now?
So right now we have SMI, we have 6. They still work. We're gonna try not to use them, but we have 6 of those. We have 2 Tobies and 2 Smart Eyes. I would like to buy a few more of either the Tobii or the Smart Eye. I think we're gonna evaluate in the coming months with some trials in the lab. And I have a feeling it's gonna be Tobies because it's mentioned here. Here's again, just a reiteration of the speed, accuracy. This is the big thing for SMIs. There's no support for that anymore. The company doesn't exist anymore. So the strengths, they're flexible. We've lots of experience with them under Tobii. They are robust, they're flexible. I think they're kind of the industry standard. They're the most common one you're going to find in labs. Smart Eye is going to be, I think, probably less of a learning curve than Tobii because it's tied to eye motions, which is very easy to use turnkey software for doing recording. And multimodal, what I call multimodal recording. In other words, eye motions is designed to integrate signals from other sensors as well. If you want to do heart rate and skin conductance and all of that with your eye tracking, eye motions just kind of does that out of the box. So you're probably gonna want to use that machine. So weaknesses, yeah, SMI out of support, old. I think Tobii, we're gonna have a slightly steeper learning curve. We're still evaluating what other softwares are going to need to do what we want to do with it. And then the Smart Eye, the weakness is you're kind of locked into eye motions. And eye motions is not the ideal stimulus presentation for certain types of experiments. If what you're showing are slideshows, videos, it's fine. If you're doing reaction times and things and more complicated interactions, it's really weak in that area. So what was I trying to say here? So I guess this is just like your workflow. Study design, participant recording, and then data analysis. So we'll kind of step through this quickly. So the stimulus display software connects to the eye tracking server and allows you to design experiments in either storyboard fashion, so iMotions or ProLab, which comes with Tobii. You're building a timeline or storyboard of like slides. As long as they can handle very simple randomization, but nothing too complex. If you need something a little more complex, then you can use something like PsychoPy or Psych Toolbox and connect externally. I think Tobii's going to be stronger there than the Smart Eye. Every experiment. Go ahead. Yeah.
Oh. So the basic experiment, like you would like present some stimuli, and then connect, and then it's matched with the software.
Right. So I think I have a little kind of a diagram here. There's like 2 different setups that you might want to use. So depending on the software, you have your experiment software, say here on the left, that is, again, the interface with the participant, showing them stimuli, it's recording responses. It's connecting to the eye tracking software, and the eye tracking software might be streaming samples 250 times a second to the software, which is then writing that data. That's one way of setting up eye tracking. You can do that. That's the more bundled turnkey things like eye motions. That's basically what it's doing. And then analysis is also built in. So that's one of the easier ways to do it. Slightly more challenging way of doing things, but more flexible, is your experiment software is really just handling the stimulus display and interaction with the participant. Your eye tracking software is independently recording data. What you're sending are timestamped events, stimulus onset changes, things like that. Maybe reaction times or decisions, and it's going to. So then your data lives over here. So depending on the software you're using, we can talk about specifics, like if you're doing the most flexible stimulus presentation software that we've been using is like PsychoPy. We've done a lot of that. That's written in Python. You can write your own code, so it's very flexible. You can do more or less anything you want. It can work in either mode. This one's a little easier to set up, but at the end of the day, you have their version of the data stored in like an HDF5 file that you have to then figure out how to analyze. So depending on your analysis tool, you need to just. So I think part of my point here is you want to think about all of these steps, because how you're going to do your data analysis informs how you're going to design your study and how you're going to set all of this up. This is just an example of calibration. So every step, every study begins with calibration. That's really how you map an individual's gaze to the pixels on your screen. And this is just an example of I looked at these 5 points and it's redisplaying to me in little dots where it measured my eyes. You can see there's a little noise in there, but that's actually pretty good. And there'll be statistics on this, how accurate that was, and those should be stored with your experiment data. So tracking data, so the lowest level, it is just a time series of eye movements, 250 or 120 times a second. They'll be in like angular units or pixel units. They can tell you a lot about an activity, the dispersion or again, the reverse, or how focused, say, someone's gaze is. They give you some insight into cognitive processes. And we work with most of the positional data that's anchored on the stimulus itself. And calibration is really important for that. So the basic unit of positional measurement is the pixel on our screens. Like you mentioned, so this top left is going to be 0, 0. So you'll get a date. So the initial data set you get will have X, Y in this coordinate system. Again, sampled at whatever your sampling rate is. And if you were to plot that, say draw that over in 2 dimensions over your stimuli, it might look something like what you should see below here. A very squiggly kind of thread of just kind of noisy eye movements over, in this case, some text, which is difficult to use for analyses and statistics. So what we tend to do is then run that raw data through an event detection systems or event detection algorithms. What they do is they try to identify where fixations are probably occurring, most of their fixation first, and then they move through the samples and the next fixation. And then anything in between is a saccade or a blank. We can kind of get to that. But eye detection is important, or eye event detection is important. So here's, for example, something like a scan path of that same data that we saw before. So kind of moving from this squiggly mess to something that's a little easier to handle. Here we have fixations over time that have durations, and then lines in between them. And now your data is getting closer to something that you can work with. And it's an important step in your whole process. It's this event detection step. Here it is, another example. And just examples, say, of what the difference in that data, how that data might look. So on the left, here's just a demonstration example of what raw data might look like for a single eye. Just timestamps, XY coordinates according to pixels. After event detection, what you'll get is a line per fixation. with the duration in milliseconds and the center of that fixation. Most of our positional eye tracking analyses will use fixation data like this. And there's a number of algorithms that you could use to do that. There are different ones are plugged into the software. You could write your own. They're not that hard, and that's something you want to think about early on in your study. I know we're getting toward the end, so I'm going to kind of move quickly here. So one thing about those fixations is you're still working in pixel space. So there's a lot of pixels on the screen. What you might want to do is sort of start binning that into areas that you might define. So we use you'll see the phrase area of interest, or AOI, used all the time in eye tracking research. So what you might do is say, draw a rectangle somewhere on your screen, and rather than looking at just the pixel that somebody focused on, is that pixel inside this area of interest? Another level of aggregation up. So they can be simple grid. You can just grid out the whole screen, in which case really what you're doing is sort of downsampling the whole pixel space. But usually what you do is you let the stimuli itself decide what AOIs make sense to you. So for example, back to our pedigree ad, I drew an AOI square around the birthday boy's face there, and then one around the ad logo and text. And down here we see some few examples. There are more AOI statistics around this, but here's some common AOI statistics. So in this case, we're looking at 12 participants. How many participants saw that, like entered that area on the screen? What was the average dwell time within that screen, like a summation of the fixations in there? How many fixations? The time to find it. So say from the onset of the stimulus, how long did it take them to get to this one, to get to this one? Total number of glances, total number of revisits. So like how many times did they look at this, leave, come back to it? Might be a measure of comprehension or interest or something like that. So yeah, so now we've gone from say files with thousands of lines to just a few summary statistics that tell us a lot about what's going on in this scene.
Robert,
Yes.
does it even make sense to draw inferences from the aggregate number? Because there might be some people who look 5 times to the person, right? And others just once, and then if you aggregate, that's misleading, right?
It could be. And
The
actually,
outliers are-
often we look at the individual statistics within this,
Okay.
but for this, I just showed everybody. But yes, you're right, but usually you look at it per individual. And then here's, just to give you a bigger picture of that, here are all of those individuals represented as scan paths over that. And so that you can see how the AOIs just give us a lot more, map those fixations onto meaningful parts of our stimulus. So quick reporting guidelines when you're publishing, people often don't do this stuff, but always mention the eye tracker model, the specifications, what the environment was like, the lighting, did you use head stabilization, all that calibration data loss, for a single participant, what percentage of time did you actually have eye data or not? And how many participants do you have to exclude? You often lose participants due to problems with contacts, problems with glasses, problems with all kinds of issues. And what algorithms did you use for eye events? What stimuli did you use, and what AOIs did you use if you did them? There's a lot more we can get into. Transition matrices are an interesting way of sort of linking the scan path idea with AOIs. So the order in which AOIs were visited, and then you can look at, say, 2 individuals and do similarity metrics between them. There's heat maps, focus maps. I mentioned the similarity metrics. And what did I write here? Oh, so similar categorization. So one thing I kind of glossed over is how tightly your stimuli are sort of tied to the eye tracking and how alike they are between runs. If you show the same people the same images and for say the same period of time, your analyses are very straightforward. Moving away from that, if you're say using video, then the more dynamic the stimulus, kind of the easier it is to set up your study, but the more work you're kind of backloading into your whole workflow. And then I didn't really talk about glasses, but if you're using say eye tracking glasses, then everyone's stimulus is not only dynamic, but unique to that person. So again, your analysis gets more and more complicated.
People can use glasses, their own, like, people,
So you can think of like the easiest way to adapt something you're doing to eye tracking is just throw glasses on a person. You're really setting yourself up for some challenging analysis at the end if you do that though. So that's the kind of spectrum I see of, are you going to do that work up front and redesign your study so that it's easier to analyze with eye tracking data, or, you know, just wait until the end and figure it out. I think this is kind of the end of it. I just wanted to probably make this public, so I wanted to put my email address here. If you're interested in eye tracking, this is a really helpful. This book is a really helpful guide. And then here's an article for things you should report on when you're using eye tracking in your studies. And my watch is not on.
That's good.
It's like showing me a message. We have 5 minutes if we want to talk
Can you
about
say
anything.
about sample sizes, like how many participants would you run, and Robert, like you just described,
Right.
like what it takes to actually do it in the lab?
I mean, you have to think about what your questions are and what, you know, and to Max's point, like what, you know, how much heterogeneity do you expect there to be? From that, so, I mean, the last study that we published with eye tracking data had a sample size of 72, we lost a few, I think we had 77 and we ended up losing like 5. That just couldn't be calibrated, like lost them at the point of calibration rather than like midstream. That's decent for the kind of things that we were looking at for around patterns and other subsequent timing behaviors. Yeah, I mean, it gets murkier as you get into more complex kinds of questions where there might be more individual differences. So yeah.
Like anything, yeah, it's a function of your effect size, but then you do hit up that against that practical limitation that is just slower to collect eye tracking data.
So like,
But
a participant walks in, we need a WBL staff person to set them all up.
Yep, exactly. And that's the main limiting factor.
And then it takes half an hour per person, sort of thing, or
That all depends on the length of the study. I mean, the calibration and validation steps add a few minutes.
okay.
That's not really the limiting factor. It's really that it's like one person per room, and we need an RA to really set them up. And then we only have so many devices. And then there is a fair amount of attrition due to data loss issues.
And you do probably repeated measures as well, right? So to increase the power.
Repeated measures, it depends, like, right, so you do, like, if within sample.
Yeah.
Not necessarily, because if you're getting this, like, because people's strategies change, as they are more familiar, so again, that really depends on the questions that you're asking. You should come to my class. I do the whole semester. So. Robert, you did a really good job in 40 minutes.
Thank you. It was a little bit of a whirlwind. I hope it made sense.
Super,
Okay.
Yeah, any other questions?
I'm happy to help.
I also
with
would-
strategy to
yeah.
do with people that want to.
Great.
And if anybody wants to do some hands-on work with this, get in touch with me, because I really need, we can only go so far, and with these, just as you know, setting up in the lab and doing demo runs, like if you have ideas to bring to us, let's like set aside like an afternoon and do like a hands-on workshop.
Great.
And think about think about what you want to use, whether that's like Python or MATLAB, or whether that's like which eye tracker you think would be best for you. Just try to use this to guide as a guidepost.
I just wanted to, anybody on Zoom, if you have a comment or question.
We do have a chat. Let me see what that
This
is.
is Sean's.
Oh, okay. I will not click that. Pull it
Great.
on.
Great. Thanks so much for being engaged and coming. If you have ideas for other ways that we could be supportive, or ways for us to connect scholars doing eye tracking with people that want to start off and you know, get the people with it, you know, let us know. Great,
Thank you.
oh, this is great, thanks so much, this will, you sort of really distilled a lot, and I've been curious.
Yeah.
About this, I think I saw one 30 years ago, and
Come a long
the
way.
technology looks just the same.